nanoHUB.org.
Online simulation and more for nanotechnology.
All right. Hello, everyone. Thank you for joining us today for our event, Hands-on Teaching with Jupyter Notebooks and nanoHUB. With us today, we have Dr. Michael Reppert. He is an assistant professor of physical and theoretical chemistry at Purdue University. In 2019, he began his own research group in physical chemistry at Purdue, working to develop both experimental and computational tools to quantitatively understand structure and spectrum relationships in biomacromolecules, particularly photosynthetic proteins. So thank you for giving this presentation today, Dr. Reppert.
Thank you, Amy, for the introduction, and thank you, Tanya, and the team at nanoHUB here for the chance to talk today. So yeah, I wanted to share a little bit about what we've been doing here at in my group and in my the classes that I teach, how we work to integrate nanoHUB content into those courses, and how that's been useful for us in our applications, mostly teaching physical chemistry, but usually with a sort of biology focus. Without further delay, I'll get started. So before I go into content, I wanted to explain a little bit about my background and sort of how I got to where I am now. So I started out in research as a physical chemist. My PhD is in ultrafast spectroscopy. I work in an experimental group at MIT at University of Chicago doing ultrafast vibrational spectroscopy. And then after my PhD, I went off and did a postdoc in open quantum system theory. So very much pen and paper, fundamental questions, very much outside the lab. And in my group here at Purdue now, we try to kind of integrate those, integrate all of those features together. So we write open source software for analyzing protein structure and dynamics, especially focused on photosynthesis and understanding processes like energy transfer in chlorophyll proteins. But to keep things sort of tied to the real world, we also I also run a molecular biology lab. So I like to say we're sort of a sham molecular biology lab in that I'm very much a physical chemist, but with some molecular biology training. So we produce, we grow photosynthetic organisms, cyanobacteria, and produce photosynthetic proteins through recombinant expression. And all of that is sort of to back up the theory work that we do. So we try to develop software to make predictions about energy transfer and photosynthesis. And then we actually try to go to the lab and create those the various constructs that we've designed and try to see if they actually behave the way we think they should. So we're very much trying to test those computational packages and theoretical developments sort of in real time. The reason that I mention this, so this is not a research talk. It'd be more comfortable if it was a research talk. I'm a lot more used to talking about research than education, but this isn't a research talk, but I mentioned this just by way of background to say this is kind of where I'm coming from in the way that I teach. It's very important for me to, both in the students in my group and in the students that I teach, I want to be able to help them build really strong conceptual connections between the sort of math side of things, the open quantum dynamics that we do in the research group, and then this real world of what happens in the lab, and the kitchen and the rest of daily life, especially in the context of biology. So that I have found a lot, both in research and in teaching, that there's often a pretty strong disconnect between mathematical models of the world and the sort of basic physical laws that we teach in physics classes, and then the applications in or connections to biology and the life sciences. So that's sort of a real passion for me, and somewhere a place that I've spent quite a lot of time thinking about how to get people connected to connect those two things. And in what I've found so far is that one of the big challenges, I think, to doing that, to building those kinds of connections, is to get students to sort of slow down a little bit and think more carefully about their assumptions and the way that they think about math and physics and biology. From the physics side, physicists tend to sort of ignore biology as kind of, I don't know, boring or just a lot of memory work. There's a famous story about Richard Feynman, a very famous physicist, who is kind of making fun of biologists, or making fun of, I think it's veterinary students for memorizing anatomy, because he thinks, well, if it's in a book, you can just look it up. I can find a map of a cat in a book somewhere. I don't need to memorize it. I just need to think about it carefully. Which is all well and good if you're a physicist, but not so much good if you're a veterinarian, working on a cat in the operating room. So physicists, I think, tend to kind of be dismissive of biology as just a lot of memorization, and then biologists tend to be a little hesitant to delve into the physics and the math that's really fundamental to thinking about physics. And it's often very hard to get the two sides to sort of cross-pollinate with each other and think about how the math applies to the physics and how that biology should actually inform the physics that you're thinking about in the lab. So that's getting people to sort of stop and think is one of the big challenges, I think. And then finally, a way that I found, so this is both a practical tool and a means of achieving that end of getting people to slow down, is getting students to do a little bit of programming and works with some molecular simulations. This tends to be an easy place to force people to slow down because they have to stop and think about how to write a computer program and then think about the basic physics and the biology application that they're trying to model through programming. And then this is also, of course, works out well for many students in the long run in that programming skills are becoming an almost must-have commodity for in a lot of fields of work. So I also try to get students thinking about programming both as an educational tool and then as a resource for them in their work later on. And then finally, of course, it's really important, I found, to keep people engaged in having some fun with these kinds of concepts, just so they don't sort of tune out and go off in some direction. So what I'm going to talk about today is how I've used nanoHUB, and especially Jupyter Notebooks hosted on nanoHUB, to try to work toward these goals in both the college courses that I teach, graduate and undergraduate, and then also in science outreach projects. Okay, so what I'm going to do today, just to give you a preview of what's coming, I'm going to talk a little bit about background material, define some of these terms. What are Jupyter Notebooks? What is nanoHUB, for those of you who aren't familiar? And then I'll, in the middle third of the talk here, I'll talk about specific applications in different courses, both undergraduate and graduate, and in science outreach. And then last, I'll give some ideas that I have about the easiest way to get started with these kinds of writing notebooks for publishing on nanoHUB, how to actually create the notebooks, get them installed, and get them working properly on nanoHUB. That process is fairly easy, but it's maybe not quite as obvious as one would hope. So I'll have a sort of very brief set of tips and tricks for how to get that working. Okay, so first let's start off with background. What are these terms Jupyter Notebook and nanoHUB mean? So if you already know all of this, feel free to check out and tune back in about 5 minutes. But for those who aren't regular Notebook or nanoHUB users, it's actually a little bit hard to define Jupyter Notebook in simple terms. Basically, Jupyter is a software package that lets you run Python code and other types of code, R and Julia, I think maybe a few others now. But it's a software package that lets you write and run code in your web browser. So it's in a normal Jupyter Notebook installation, you have to actually download software, install it on your computer. And then when you open up the interface, it'll actually open in your web browser, Chrome or Internet Explorer or Microsoft Edge or whatever. Mozilla Firefox, you'll actually open it in a web browser and you'll run the code in the web browser. So it actually looks like you're running it online, but you're not. In a normal Jupyter installation, all of the software is installed on your computer and you're running it on your computer. It's your computer's processing power that's executing the code. Okay, the nice thing about this framework is that it's a sort of simple looking interface. You don't have any complicated Microsoft Basic environment. You've got a simple web browser window, and you can write code in these little cell boxes here. And then when you execute the code, the output appears down below, and you can write things like these, like this visualization app illustrated in the window. This is just from the Jupyter Notebook webpage. Okay, the notebook itself is the document that you write. So this is a file that you create. Again, looks like it's in your web browser. But you can create the file and save your code, save the output from the code directly in that Jupyter Notebook, and then you can open it up again later and run the same thing. Those usually come with the extension .ipynb. That stands for Interactive Python Notebook, because the main engine that's running all of this is Python, at least most of the time. You can also switch to other languages, but Python is the default usually. Okay, so this is long story short of what is a Jupyter Notebook. The interface is nice, but again, you have to install things on your computer in order to run it normally. This is where nanoHUB comes in. So nanoHUB is this science gateway, if you like, that provides, for my purposes, most importantly, this open access Jupyter Notebook environment where you can log into a website. They have the Jupyter software installed on their computer, on their servers at nanoHUB, and you can run the notebook in your web browser exactly as though you're running on your own computer, but you're using their compute power to actually do the calculations. And this is nice for a couple of reasons. One is just that, if you don't have a powerful, if you're running on your laptop, you may run into computing issues. You may not have enough power to execute a difficult code. But more importantly, I think for most purposes, it's nice to not have to worry about installing anything on your computer. This is especially important for education applications where you don't want to work through with 100 different students in your class how to install Jupyter on your on individual laptops. Trust me, I've tried it, and you don't want to do that. So this is where nanoHUB really comes in handy in education contexts, is that you can have everybody use the same site, everybody has access to the same materials, and you don't have to work through on a case-by-case basis, helping people figure out how to run Python code. In particular, in addition, then nanoHUB has this app mode interface. So there's a little button you'll see that says either enter app mode or edit app. You can toggle back and forth between these different modes. And when you're in app mode, all of the code gets hidden. So normally when you run a Jupyter Notebook, you can see both the code, this little gray box here, and the app, the output of that code, the app that you can run down below. On nanoHUB, there's an option to enter this app mode setting where all of the code disappears and all you see is the GUI, the graphical representation of the simulation or the plot that you're trying to make. And this is nice for making sort of point-and-click apps where you don't want your students to have to worry about accidentally deleting all the code or modifying the code. They can just run the simulation, click buttons, and move on with their lives. Okay, and then finally, as I mentioned, there's these computational backend resources. If you want to run a molecular dynamics simulation, that might be hard for a student to run on their laptop, even if they had the software installed. On nanoHUB, things like this are relatively easy because different member institutions, including Purdue University, but I think there's at least a dozen other schools that do this also. They donate computational resources to nanoHUB so that you can run things like molecular dynamics simulations, again, directly in your web browser, but the compute job is actually going to somewhere else in the US. I think they're all US-based institutions at this point, but the simulation is actually going off to some other institution and then coming back when it finishes. So you can get a lot of compute power for free, and anybody can do this. You can just register for an account on nanoHUB, and then you get access to these resources if you know how to find them. This is a pretty big system. There's upwards of a million monthly, upwards of a million annual visitors to the site, with on the order of 200,000 monthly visitors. So there's a lot of people using the software or the interface. It's possible to get pretty good traffic for your apps. Okay, I've already run through most of these, but I wanted to again highlight the sort of advantages to running Jupyter Notebooks on nanoHUB specifically. So you get the real point is you get all of the advantages of Jupyter Notebooks, which is an easy to use interface for doing Python programming without software installation. This is the really big thing for education, I think, is that if you're writing software for your own personal use, it's okay to have to install stuff and then you run it. But when you're trying to get the software working for 100 students in a class every semester, that can just be real complicated if you're trying to do it, make each student install software on their own. It's much easier if you can distribute data to different users on a central site. And that's what nanoHUB is really good for, in my opinion. Okay, I want to emphasize that it's easy to share files on nanoHUB in one direction. So it's easy for me as an educator to push files out to students so that they have access to them. There isn't a native interface for students to push files back to me. Okay, so when I'll talk later on how I use these simulation apps where students have to edit the app and then submit some homework where they've done some coding. They don't do that submission to me on nanoHUB. So they would write their software on nanoHUB, but then they actually download the file and send it to me via some other secure file system. So nanoHUB does not, as far as I know, have any kind of FERPA compliant secure system for sharing files or grades. That part should be handled separately, at least from my perspective. Okay, as I mentioned before, you can keep your code visible or you can hide it so that students don't have to deal with it. You can use this so that students can either have to program, in which case they learn some programming along the way, or they can just use it as an application where they're just users and see the output. And again, there's access to compute power that I think you almost can't get anywhere else. Okay, so that's my sort of whirlwind introduction to nanoHUB and Jupyter. So now I'm going to give a few illustrations of how I've used that, how I've been using those tools here at Purdue, integrating those into my courses. So the first example I'll give is for a physical chemistry course that I teach. I've taught this now twice and teaching it again in the spring. This is the title of the course is just physical chemistry, but it's a one-semester survey course that's specifically targeted at life science students. So this is specifically for especially pharmaceutical science majors, biology majors, and biochemistry majors, who are usually coming in with not as strong of a mathematical background as our sort of core physical chemistry students, but they still want, a fairly deep conceptual understanding, at least, of the kind of thermodynamics and statistics forces that determine problems like protein folding in biology. So protein folding is an example I like to use a lot because it's relatively easy to see the connections between the statistics of, what are the populations of different protein conformations, and then the biological function of, the protein has to be in this particular conformation to perform its biological enzymatic function, for example. So in this course in particular, we're shooting a lot for conceptual understanding, although we do actually cover the math in fairly great detail. We just work through it a little more slowly than I would in a PChem course for PChem students. But especially the emphasis here is conceptual, and specifically what I needed for this course, most of the course I teach as a standard, 3 days a week lecture, 1 day a week recitation, standard chalk talk PowerPoint course. But I wanted, especially for building these connections between math and statistics and biology, to help students get a feel for how statistical forces and physical driving forces lead to protein folding. So on nanoHUB, then I built this standalone app where students can log in and sort of play with a toy model of protein folding, where they build a peptide, a short sequence of peptide. I think they can put in up to 15 amino acids. And then the code will go then and calculate what the probability for the system to be for that peptide to be in a particular conformation is at 298K. And it'll also show then the length of the chain, the total length of the peptide is a function of temperature, and the partition function, the normalized partition function, which counts the number of thermally accessible states as a function of temperature. This is a very much simplified model, so there's only 4 amino acids: alanine, glutamate, lysine, and proline, and they have very coarse-grained properties in that. So alanine has alanine is hydrophobic, glutamate E is negatively charged, lysine K is positively charged, and then proline likes to form turns in one particular direction. Okay, and so that's the basic idea. And then what the app is doing on the back end is going in and calculating, given the sequence of amino acids that are entered, then the app goes in and calculates all of the different possible conformations for that particular peptide in this 2-dimensional grid, and then calculates what's the probability based on the energetic interactions between the amino acids, calculates the probability for the peptide to take any one conformation. So if you put in a sort of complicated sequence like the one that's being entered right now, you can get unique protein folds. So you'll see this one forms a sort of collapsed kind of triple hairpin-like structure where you've got proline kinks forming these turns and a hydrophobic alanine core. Or if you put in this case, just polyalanine now, we've got a bunch of alanines together. This forms an unstructured but almost entirely collapsed structure where the protein is always collapsed into a lump. I use this when we talk about hydrophobic collapse of proteins. Or if you put in a bunch of glutamates, these are all negatively charged. The protein comes out flat, almost entirely flat. Again, not with any unique structure, but with many different extended structures, reflecting the fact that those glutamates are all repelling each other and trying to stay as far apart from each other as they can. And then in homework assignments, I'll have the students do things like design new peptide sequences that have particular properties, or fill in the blank, which amino acid is missing to make this fold in a certain way, or study as a function of temperature. Why does the polyalanine chain length increase as a function of temperature, whereas the polyglutamate chain decreases as a function of temperature? Because in the one case, as a function of temperature, it's better for the alanine it becomes easier for the alanine to extend as the temperature goes up, but it also becomes easier for the polyglutamate to condense as the temperature goes up. Those conformations become more accessible. So this is just a single standalone tool that my students use and has worked out pretty well for illustrating these concepts. And students tend to like the sort of gamified nature of it, trying to figure out, trying to guess what amino acid they have to put in or design sequences. And it seems to do a pretty good job of, especially illustrating those concepts, things like the partition function, the number of accessible states that tend to be otherwise kind of hard for students to wrap their brains around. For the other physical chemistry course that I've taught here, this is a physical chemistry lab. I wanted something a little bit more complete, so not just a plugin that they would use once during the course, but I wanted to build a home page for the lab where everything would be consolidated into one place. And the motivation here is that, so we're working now here at Purdue at updating some of our lab resources, and one of the problems that I was facing is trying to consolidate a whole bunch of scripts that have been developed over the years by different people to teach different labs into a single collected place where students can access their resource, access their instructions, their lab instructions, enter data, save data, plot data, and learn a little bit of programming and data analysis along the way. So on nanoHUB, I built a sort of one-stop shop homepage where students can both get their lab notes. So you'll see here if they click on one of the links on the schedule, it'll take them to a separate nanoHUB page that has the sort of main handout for that lab. We can do things like embed molecular viewing software here so that they can view structures of the molecules that they're dealing with. They can click on links to open lab instructions. So this is the specific set of instructions that they'll actually do in the lab. So students have access to computers during the lab session, so they can go and actually follow along online as they're collecting their data. And then when they're done, they create a new notebook. So it copies a template here, actually. They can open up that template and then start filling in that template with the data that they've collected in the lab. And ultimately, it'll give them instructions for how to do their homework. So you notice there, the student just clicked an edit app button that was showing up here. This is the app mode button to get back into app mode. This is now Python code that they can modify and execute. And this is where they'll load their data into the lab and then modify the code to plot it correctly, build least squares fits, type in answers to questions, things like this. You can type in either plain text, or in this case, this is an example using LaTeX, where you can get nicely formatted equations. But there's a lot of different options for how students can use this. And the way this actually worked out practically is at the very beginning of the course, when students are coming in, most of them have no idea how to do any programming. And so at the beginning, this is an example from near the beginning of the course. At the beginning, the notebooks that I give them are almost complete. They have to add like one line of code or change a file name or enter one simple command or something like this, and then the whole thing will run. And then as we go through the semester, the notebooks get less and less complete until the final notebook for this semester is just a set of instructions. There's no code when they start. They have to write everything themselves from the beginning, or copy and paste the relevant part from some other file and work it into their folder. And that's worked out pretty well. Students pick up pretty quickly how to modify the code to plot what they want. There's, for a physical chemistry lab, there's not that many different things students need to do. They need to be able to make a plot. They need to read a text file. They need to make a plot, make a least squares fit. There's a relatively small number of commands they have to learn, and then they can have the confidence that they can actually plot data and make more or less publication quality figures with a notebook like this. Give them something other than Excel to plot and interpret data with. That's where we've gone with the lab notebooks. And now everything is pretty much standalone. Everything that they get is all the materials they have access to are hosted here on this page. And then, of course, as before, when they're finished with their homework, they'll download the code and then submit that either as a PDF or as a notebook for grading through our FERPA compliant software for grade distribution. Okay, so that's what we've done with physical chemistry lab. The content for the graduate course that I teach in molecular spectroscopy is similar. So we, again, I use nanoHUB to host the homepage. The main differences here between the graduate course and the undergrad course is, in this case, I'm expecting students not necessarily to come in with more programming knowledge, because some of them, again, don't have programming knowledge when they come into the course. But I am expecting them to develop those programming abilities faster, given that it's a graduate course. So by the end of the semester, students in this course will not only be able to plot data, but they'll be able to do things like write or at least modify molecular dynamic simulations with Python code in order to be able to visualize the kind of light matter interactions that we think about in molecular spectroscopy. So in the most unique thing I think about this course is that instead of in most spectroscopy courses in chemistry departments at least, I think sort of start off with quantum mechanics. As chemists, we like to think a lot about quantum mechanics because selection rules and things like this are very important in chemistry applications. But I think there's a little bit of a gap in understanding for many students then, in that most students haven't really developed the conceptual background for how light interacts with matter at a fundamental level. And so we try to fill that in this course by giving a bunch of illustrations, both on paper and then on the computer, for actually what happens when a laser pulse comes in, for example, and interacts with it. So this is a simple example. This is just a molecular dynamic simulation, and I actually work through with the students how this code works. They actually write and modify the molecular dynamic simulation themselves. We just have dipolar particles or dipolar molecules. The blue is a positive charge, red is a negative charge, and there's a thermostat that keeps the temperature condensed down to 0 Kelvin. And so the molecules gradually assemble. Initially, they collapse into this thing that looks kind of like a dog. That's not intentional. That's just coincidence. It's how they happen to fold in the simulation. The light pulse comes in and sort of blows them apart. And then they collapse back into some other structure. So again, here's the start of the simulation. The molecules are initially randomly distributed. It's cold. The temperature here is set to 0 Kelvin. Students have control over that. They can modify any of these parameters here. And then the system has sort of reached a local equilibrium here. A laser pulse comes into the system, blows the system apart, and then it recollapses into some other structure as the simulation progresses. And there's a whole slew of parameters controlled by these slide bars up here that students can change when they run the simulation. The T pulse is the length of the time that the pulse arrives in the system. In some of the nonlinear spectroscopy simulations we do, there's multiple pulses, so students can see the effects of several different pulses interacting with the system as a function of the delay time between those pulses. The tau pulse is the duration of that pulse, how long you keep the light on, the frequency of the pulse. They get to look at things like resonance. What is it? How does the response of the molecular system change when the pulse is resonant with the molecular vibrations of the system or not resonant? The intensity of the pulse, how big of a perturbation is it? How many molecules there are? How long do you run the simulation? Eventually it gets tiring to watch molecules jiggle around. Gamma here is a friction coefficient that controls the viscosity of the solution around the molecules. And then, of course, the temperature, which affects the equilibrium state of the system. And so this is kind of, this is just one example. There's, when I teach this course, there's typically one applet like this that students work with every week. And in contrast to the most of the other examples that I've shown so far, in this course, students are explicitly meant to look at the source code for the app. And typically, they'll actually have to go in and modify the source code to make some change as part of their homework assignment, so they get a sense for how a simulation like this runs and how they could write it themselves. And then, what is the effect of various parameters on how the system works. And then they typically have one assignment like that per week. I give two lectures, two stand-up lectures every week. And then the third lecture each week is a computer lab where we work through a notebook problem like this together. And then students have some take-home work to do that they finish on their own time. Okay, and then finally, I wanted to close with those are the three sort of formal courses I'll talk about. I wanted to mention briefly here then some science outreach work that we've also done with the Purdue K-12 science outreach folks here, Sarah Nern and Stephen Smith in physics. So as I mentioned before, it's one of my passions is trying to connect biology and physics. And in my research group, we do a lot of vibrational spectroscopy to study protein structure. And so we put together a lab where students get to sort of dabble a little bit in understanding how spectroscopic information can tell you about what's happening at the molecular level, in this case, in protein structure. So we start out in the lab. Well, we start out with a lecture. I give a chalk talk for about 30 minutes about the history of protein structure determination and how we know what we know about the way molecules are oriented in space, especially in the context of proteins. And then we do a little lab work where we actually boil an egg. So we start with raw eggs, boil half of the egg, and then go upstairs to my lab and we take an FTIR measurement, a vibrational spectrum of both the raw egg and the cooked egg. And what students see then is that when you cook the egg, there's a concerted shift of one of the spectroscopic peaks in the FTIR spectrum down about 20 wave numbers to the red. So you go from about 1635-ish down to about 1620-ish. Or 1615-ish, maybe even here. So you get this big redshift in the spectrum. And then what we do is go back down to the lecture room, and students log on to computers and pull up this this AMID spec app linked in the toolbar here at the bottom. And here, students can pull protein structures from the protein data bank. You can actually enter any protein structure you like that's in the protein data bank. It'll pull the structure and then plot the carbonyl groups. So these gray and red sticks sticking out here, these are the CO backbone groups of the protein, and those are what give rise to this vibrational pattern in the experimental spectrum. And then students can see what happens as a function of chain length. So as a function of how many carbonyl groups are included in the calculation, how does the vibrational spectrum change? And what they'll see is that if they select only one chain from this multi-chain subunit, so here they're going to select just chain A, and they're going to run a calculation using just that one chain, just a single strand. What they'll see is they get a broad symmetric band around 1650 wave numbers. If they go back then and add all of the chains, you've got this much larger structure. You'll see that your vibrational spectrum shifts to the red by about 20 wave numbers and becomes asymmetric. Okay, and that's exactly the same kind of shift that you see experimentally when you go from the raw egg to the cooked egg. And this is in fact exactly what's happening in the raw egg. There is when you're cooking the egg, the proteins are getting denatured, and you go from proteins that were folded in many different shapes, alpha helix, beta sheets, but generally smaller structures in the size of one protein, into these long beta aggregates where many proteins are collapsing together and forming these big multi-domain structures like shown here in the visualization window. And that's actually what leads to that redshift in the absorption spectrum. So both computationally and experimentally, you can see that same peak of the vibrational spectrum to the red. And students are able to relate that to this really hands-on way to the cooked egg and the raw egg that they're playing with in the lab. Okay, so I'm going to stop there in terms of content, and now I'll introduce, in the time that I have left, a little bit of a sort of set of tips and tricks for how you get started writing code like this or hosting apps on nanoHUB. So the first place to start is, as I think Amy mentioned to begin with, is creating a nanoHUB account. I think she already provided the links to do that, so I won't go into that in detail. But you can go in and create a nanoHUB account. It's free, although you do have to register so that they can sort of track how resources are being used. And then go in your browser to this this particular tool, nanohub.org/tools/Jupyter70. This is the updated version. And this is where you can get access to that online Jupyter Notebook server that I mentioned. So you can log in here. If you follow that link, you'll go to, you'll be directed to a page that looks something like this. It's a folder viewer where you can see what files, if any, you've created. And here you can create new Python notebooks. So this new drop-down menu over here, if you click on this, there will be an option for Python 3. That's the sort of default installation of Python. You can create new notebooks there and start programming away. And then you can test those programs using that app mode setting that I mentioned earlier. Click on the app mode button and it'll take the code all away, and you'll just see the GUI output or the output from the code, depending on how you set it up. If for those of you who have some Python experience, if you want to create these interactive notebooks where you have slide bars and accordion windows, checkboxes, things like this, this IPy widgets library stands for Interactive Python Widgets. This is very much your friend. So virtually everything that I write on nanoHUB, I think actually everything that I write on nanoHUB extensively uses this IPy widgets library. With this library, it's relatively easy to define these Python objects that correspond to graphical interfaces, graphical objects like slide bars here. So this is an easy way to get user access or user input on, how long do you want a simulation to run? What should the temperature be? What should the viscosity of the solution be? Things like this. These slide bars or other, checkboxes. There's a whole world of widgets you can put in, buttons to click to make the simulation go. Plot viewers to or display output displays where you can embed matplotlib plots, things like this. So this is sort of a must-have resource for developing these interactive tools. Okay, and so then once you have a notebook written and it does more or less what you want it to do, then this is actually, I think the bigger hurdle is maybe actually getting it published. This is this is a place where I think the nanoHUB folks could make things a little bit easier or make the documentation a little bit better. It's not totally obvious how you go from having written that notebook to getting it published. So I'll just sort of briefly walk through the basic requirements here. So if you want to publish an app on nanoHUB, there's basically two things you have to have. So one is the notebook that you want to run. This is the thing that you want users to actually open up and see the first thing when they start your app. And then second, you have to have this middleware invoke script. Okay, so what that means is in the folder where you have your notebook that you want to run, I always call it main.ipynb, just so that I have consistent naming. So in that folder where you have main.ipynb or whatever else you want to name your notebook, you have to have a folder called middleware. And inside of that folder, you have a script called invoke, a file that's called invoke. Not invoke.txt or invoke.sh or anything like that, just invoke. And that'll be a text file that contains a command, a single command that looks something like this. So this first line here is telling, this file is what tells the nanoHUB servers what to do when it starts your app. Okay, so the first thing you want the server to know is you want to execute using the shell script or the shell environment. That's this first line here. Just leave that as it is. You don't need to change it. And then this second line, this has all the parameters for how you actually want your script to run. The most important thing you need to notice here is what you probably will need to change if you publish an app like this. Just change this line here where I have Lattice protein. This is the name of the Lattice protein app that I've published. You modify that to be whatever the name of the app is that you're writing. And then at the end here, this if you choose to name your notebook something other than main.ipynb, you'd have to change the name of that notebook. Change this to whatever the name of the notebook is that you want to run at runtime. The rest of these flags you can generally leave alone. This -u flag says that I want to use the Anaconda 6 environment. This is a particular package that has an installation of the Jupyter Notebook. But again, you can generally just leave that alone. Start Jupyter just says when you launch the app, Jupyter Notebook is what you want to start. And this is the sort of backend script at nanoHUB that actually does the starting of the notebook and running the app. Okay, so this looks a little complicated. The nanoHUB support folks are really good, though, at helping you put these things together. So if you're not sure what to do, you can enter a support ticket and say, I think this is what I need. Can you help me? Or just try to publish the app with whatever you think is right in this script. And if it's wrong, they'll tell you and help you figure it out. The other thing I should say is this file has to be executable. It has to be something that can be run by the computer. So if you don't know Linux command line, just get somebody who does to help you. You just have to run the command chmod x or chmod plus x on that invoke script. Very easy, but you have to know how to do it. Okay, and then finally, so once you've got your files all ready, now you need to create a project on. I recommend doing this on GitHub. You can also, there's a way to create a repository on nanoHUB directly. I recommend doing it on GitHub for a couple of reasons that I'll talk about in a minute. But this, again, is pretty easy. It's just you kind of have to have done it to know how to do it. So GitHub is, for those who aren't familiar with it, GitHub is this a third-party, so GitHub is a website that hosts Git projects. So Git is a software package that provides version control for programming projects. So if you have multiple people working on a programming project, you can create a Git project. And each user then individually makes updates to the code, and then Git provides a way of combining those changes or controlling the changes to the code to make sure that they're consistent with each other. If you're just writing an app yourself, you don't have to worry about any of that. You just create the project, push it to GitHub. So GitHub now hosts projects on Git, or hosts Git projects, and then nanoHUB will actually go to github.com and fetch the project that you want to publish. In order to get the code and actually install it on the nanoHUB servers. So this is again relatively easy, but it does involve a little bit of command line work and learning a little bit at least about how GitHub and Git. I won't walk through any details here. You can find all kinds of tutorials online for how to create Git projects. The main thing I want to say is that this project should be pretty simple. So you can find very complicated descriptions of how to use Git because Git can do a lot. There's many different things you can do with Git. You need very little of it to publish a nanoHUB app. So basically all you should have to do is create a project, this git init command, add some files to that project, and then commit them, and then push it to github.com. So it should be a series of like 6 commands. There shouldn't be a long, complicated process for doing this. So just keep that in mind. If you're working through some Git tutorial and it looks really, really complicated, find a different Git tutorial that's simpler, because you need only the simplest commands for these purposes. The other point I want to mention is that Git is already installed on nanoHUB. So again, Git is the software that provides version control. You don't need to install that. You can install that on your local computer, but it won't help you with nanoHUB. The Git commands that you need on nanoHUB are already there. You just have to open a terminal to get to them. So to open a terminal, you click on this new button over here, click the terminal button, the tab at the bottom, and that'll open a new tab in your web browser. And that'll give you this interface where you can type commands, including things like git status, that tells you the status of your Git project, git commit, git make, git commit, git push, etc. So all of the Git activity, all of the Git the Git commands that you need are already there. You just have to access them through the terminal. Okay, and then finally, once you've pushed all of that project to GitHub, then you just have to register the tool with nanoHUB. There's a page where you fill out information, a form that you fill out to give information about your tool, including farther down this form, there would be a place where you put in the web address for that GitHub project that you've created. And that's how the nanoHUB folks will know where to get the code when they go to install your app. You'll fill out the form and then they'll fill out the form and submit that with all of the content, and then they'll try to install and then let you know if it's installed, and then you can test, and then back and forth where you test it out, make changes, test it out, make changes until it's finally ready to be published. So that part is pretty sort of straightforward once you get it into the system. I'm going to talk about, so that's sort of the brief introduction to how to get started on nanoHUB. I want to mention 2 sort of more advanced issues that have come up and took me a while to figure out how to deal with them, specifically in the context of chemistry education. One of the problems with hosting education content on nanoHUB, one of the problems at least that I found in hosting education content on nanoHUB, is that in order to make a change to your app, you have to go through this publication process where, you fill out a form to say, I've made changes to my code, please install the new version. The support folks at nanoHUB will install the code, send it back to you to test, you'll approve, they'll publish it. Okay, so there's this back and forth process with a couple of stages that, although they're pretty fast at doing that, it's not instantaneous. You've got to have somebody else on the other end checking the updates to your code. And for simulation software, this makes sense because it's kind of a security thing, right? So they're checking to make sure there's nothing malicious in what you're publishing. Make sure that it's a reasonable project. It's not going to harm anybody. So there's some real sense to the way they have it set up. For education purposes, though, this is really inconvenient in that, if you find a typo in your lecture notes and want to go change, a T to an I, you've got to get that change approved through the nanoHUB support guys. So you've got to submit a request to change your code, get it updated, you review it, you approve it, they approve it, they publish it. It's a long process and can take a while if you want to just make some kind of small change to your system. So the solution that I found to get around that is a kind of front-end, back-end architecture where so the course materials, things like my lecture notes, these I host on what I call a back-end GitHub project. And so that's where all of the lecture notes and all of the sort of direct course materials that the students interface with are hosted. And then on nanoHUB, I publish a sort of front-end app that actually goes in and fetches that content from GitHub, that fresh content from GitHub every time when the app runs. Okay, so visually this looks something like this. So I've got actually 2 nanoHUB projects, that I create on nanoHUB, a backend project where I develop all my materials. I push that to GitHub. GitHub stores all the files, and it keeps them up to date with what I have on nanoHUB. This is my personal nanoHUB directory. And then also in that personal nanoHUB directory, I create the front-end project. This is the app that actually gets published. And that provides the interface where students open up a notebook and then it goes and fetches data from the back end. Again, I create a GitHub project associated with that project, that front end, and then I publish that. This is where we go through the review and approval stage with the nanoHUB folks. That front end project gets published as a standalone app. And then when students launch that app, it automatically fetches from the GitHub server the backend, the content from the backend project. And so because all of the content is hosted in this GitHub project that doesn't go through the review process from nanoHUB, you can make changes to things like lecture notes without having to republish the entire project every time. So it's kind of a workaround to the difficulty of having to republish every time you want to make a change to your notes or something like that. Okay, so that's a relatively simple solution that works pretty okay. It takes a little bit of figuring to figure out how to get it set up, but it's not too hard. And you feel free to copy some of the at least two of the apps that I've shown here, the molecular spectroscopy app and the 373 app run this way. So if you want examples, feel free to go. Those are open source. You can download those and take a look at the source code, and you'll see how it works to do the fetching and the cloning with the fresh content. Okay, the second wrinkle I want to talk about is, and I'll try to be brief because we're getting toward the end here, has to do with data storage. So by default, when you run a nanoHUB app, other than the Jupyter Notebook app, when you run other nanoHUB apps, they open in this sort of sandbox directory that gets destroyed after the user leaves the system. So when you terminate your session, all of the data that you may have created in that session disappears. And this is okay if you're doing like a visualization simulation, like the Lattice Polymer app, where you just want to plot things and then write down your notes and then disappear. This is problematic if you want to have your students edit notebooks and then save those notebooks and then submit them for grading later on. So you either have to, if you want to do something like that, which I do in most cases, you have to either make sure that students transfer those files to a permanent location before exiting. Again, in a 100 student class, there's almost certainly going to be somebody who forgot to do that, right? So this is not a great solution. Either make sure that the user transfer files, or you can work with nanoHUB support folks to change that invoke script that launches your app, so that either it launches in a persistent folder, so it launches in your home nanoHUB directory, which is saved from session to session, or so that when the students run your code, they're forced to copy the code before they can open it. So you can actually do that on the back end. That's what's happening in the window here on the right. When the student fills out their names here and hits the copy exercise button, and then the open button, that copy exercise button is actually directly copying the data to the student's home folder, the student's persistent nanoHUB folder. And then they can go in and by the time they get to the notebook, it's already been copied and stored in a safe location. So there's a few different ways to get around this. Just be aware, you do want to get around this. You want to find some way to make sure that the student's data gets saved permanently and doesn't just disappear when they log out of the system. Otherwise, you get very unhappy students, trust me. Okay, so finally, that's kind of all the new content I wanted to talk about. I'll just conclude here. I found nanoHUB a really nice place for hosting these Jupyter notebooks. I think most importantly, there's a lot of features that are nice. App mode is really nice, but the most important thing is just that there's no installation necessary for students. That's a huge enabler for getting students actually actively working with this kind of environment. Anything that requires somebody to install something has inherent limitations in how large scale you can go with it in curriculum development. So it's a nice place for Amy's mentioning here that each student does have to have a nanoHUB account. That's correct. So students will have to create nanoHUB accounts and then individually, and then they can get access to the resources. But in any case, it's an easy way to distribute files to students and put together these both visualization and programming tutorial tools to get students really sort of digging into curricular content. Challenges here. So there's the sort of obvious one. You have to know. So you can make a notebook such that, or a notebook app so that students don't have to have any programming knowledge, or you can have them develop that programming knowledge as they work. The educator, the instructor is going to have some programming knowledge to know how to put these things together. Although this is relatively straightforward programming. So if you know somebody who knows how to code, they can probably do it if they want to work with you to develop an app like this. Especially in the sciences, maybe people aren't so familiar with those Python widgets, but they're pretty easy to get to. They're pretty easy to work with. So a little bit of investment will take you a long ways. If you already have some Python background. And then the two issues that I mentioned at the end there, this workflow with publishing and updating content is a little bit clunky. but there are workarounds for it. And then just watch out for data permanence. Make sure that you're copying and saving files at some point in the working of the app. And then finally, I'll close with the folks who helped with these projects. My TAs, Jacob and Safa, worked with me in the Chem 372 development. Paul Bauer, our lab instructor here at in Purdue Chemistry, and John Hood, another faculty member here in the department, worked in the Chem 373 development with me. All the students who suffered through my early Chem 676 experiments, figuring out how this all worked. Thanks for all your patience in figuring that stuff out. And then for K-12 outreach, Sarah Nern and Stephen Smith, who helped with this egg lab that I discussed. And then on the nanoHUB end, I want to especially mention Stephen Clark, who's done a great job going back and forth many times in figuring out how to make some of these more complicated simulations work out, or complicated interface questions work out in the apps that we publish. And finally, thank you for your time. I'm happy to take any questions I can answer.
Great. Thank you so much, Dr. Reppert. That was a great presentation. All of us at nanoHUB are always happy to help and work with people individually if they need any help with deploying anything on nanoHUB or in your classroom. So you can also reach out to us via our contact us form on the website, or you can send me an email. You should have that from all the WebEx invitations from this event, and I can put it in the chat too.
Okay, great. Yeah, so for Marcy, the question is writing a code-specific learning objective. Writing a code-specific learning objective in undergrad PChem. I see. For the college as part of the PChem major or as part of the objectives for graduate physical chemistry? Yeah, good question. So how does programming fit in terms of the learning objectives for these different courses? So the College of Science does have a programming requirement. From talking with students, I've found that many of them, so that the CS course that they take for undergrad programming, a lot of them don't find that really helpful in the sense that it's more targeted at sort of broadly at programming as a concept rather than programming in the context of data science or chemistry. Here in PChem, we're trying to work. I don't know that we've updated the learning objectives formally. But we are definitely working with it as a learning objective to try to give students an understanding of how to do programming in general. That's not a PChem specific. That's not a PChem specific learning objective, but it's a very PChem important learning objective because to actually do PChem these days, you really need some ability to program to visualize and work with data. Yeah, and for the graduate physical chemistry course, I do specifically have it as a formal written learning objective because in order to do molecular spectroscopy, you really have to know how to program if you want to do that at an advanced level. Yeah.
Looks like we did get another question that came in. Could you please summarize the Python skills students are expected to have by the end of the quarter? Sure.
Yeah, so again, this varies with the course. So with the graduate course, we try to make sure that they are able to actually, maybe I'll start with the undergrad course. So for the undergrad course, it's a relatively restricted set of Python skills. Mostly we want them to be able to import data. So basically know how to use numpy.load text. Or there's an equivalent pandas module. But there's some simple commands where you can read, comma delimited or tab delimited text files to import data into your notebook. And then to be able to plot that data, do something like add a least squares fit line to the data, or maybe something a little bit more complicated like an exponential fit. Know how to use a for loop to import. If you want to import 10 different files, we don't want them to have to sit there and program that in 10 times. So they'll learn how to program a for loop so that they can loop over file names and import those into an array or something like that. And then plot the data using matplotlib so that they can actually visualize and interpret what they see. So in the undergrad course, it's primarily reading data, plotting it, reading data efficiently using something like a for loop if necessary, and then adding things like best fit lines, and maybe doing some simple follow-up calculations. Like in one lab, we make them calculate the energies for a particle in a box using an equation embedded in the text. So relatively simple stuff like that. Basic Python syntax, especially with plotting and importing data. In the graduate course, we go a little bit farther. So all of that is included. They have to do all that as a sort of basic level. Many of them don't know that coming in. So they pick it up along the way as they go. I do the first week of the class. I actually spend in the graduate class. I actually spend the first week just doing a Python tutorial. So we actually don't do any molecular spectroscopy in the first week. We just do Python, and then we move on to the actual molecular spectroscopy. So we give them a sort of a quick jump into Python, and they learn all of that data plotting and processing, and then they also learn about things like Fourier transforms as part of the processing data, I guess. And running some simple molecular dynamics simulations, so they get a feel for how you actually write molecular level simulations in Python. And that gets into a lot more advanced stuff like updating plots on the fly and integrating or molecular dynamics integrators, things like that. But it still also builds on those sort of basic Python syntax plus some data processing and simulation tools.
All right, great. Thanks, Dr. Reppert. I think that answers all the questions that came in. Thank you so much for your time today, everyone, for joining us, and Dr. Reppert for presenting.
Great. Thank you, Amy. And if you can come up with questions later, folks, feel free to contact me. I'll be happy to talk offline as well.